One of the more common.
Okay, here we go. Okay, so where have we been? What did we talk about last time? So last time we were talking about the process of oogenesis, building an egg, making an egg. And we talked about making a sperm. That's pretty easy compared to building this egg. Building the egg is quite a complicated process, unfortunately. And I think you've had this in histology, how to build an egg. Kinda, some people are, okay. Well, we'll definitely know how to build an egg after today. So the whole story started about week 4 after folding occurred, which we'll get to, but a yolk sac forms, and the walls of the yolk sac give rise to these weird cells, and they migrate out of the yolk sac, and they start piling up here on the back of this long tube right here. This is called the gut tube. And where they pile up, we said that's called the gonadal ridge. And this is the very first, the roughest form of an egg right here. It's called the primordial germ cell. These are diploid. They're 2N, 46 chromosomes. We haven't reduced the number yet. We will, but not yet. And not all of these cells go on to become the eggs, because where do the eggs sit in the biological female? In the ovary. We don't even have an ovary yet, so we have to make an ovary, which we won't go into that story, but some of these eggs don't become, or some of these primordial germ cells don't become eggs, they become the ovary. And some of them become the eggs that sit within the ovary. Right, so that's everything I just said. So some of them become the ovary, some of them become these primordial or primitive germ cells. And we said that these germ cells, they will grow through mitosis and get bigger and bigger. And once they get big enough, you don't call them the primordial germ cell anymore. We already have a name change, and that's called the oogonia. The oogonia, or oogonium for singular, oogonia for plural. Okay, so there's our rough egg. It's still a naked egg. It has no protection around it. We need to make some protection for it, so we're going to do that here. Then importantly, we said that these oogonia, they start the process of meiosis. And meiosis 1 is that, what pops in your head when I say meiosis 1? What's that magic word? What type of a division is that? It's a blank division. Meiosis 1 is a blank division. It starts with an R. There you go. It's a reduction division. So if we make it through meiosis 1, we're going to half the number of chromosomes. So we're going to go to 46 down to 23. I always think crossover as well, because that's where the mixing of life occurs. All right. So we said, strangely enough, and for reasons we don't understand, we go through interphase just fine. We get to prophase, we cross over. What comes after prophase, by the way? Metaphase. We don't make it to metaphase. After crossover, for whatever reason, meiosis stops. It gets stuck and it freezes. And so we're stuck in prophase 1. Right after crossover has occurred. So we already have a mixing going on. All right, so let's continue the story. And I try to keep this little recap down here because it gets complicated. So we started with primordial germ cells. They got a little bit bigger in size, became oogonia, and then they turned into a primary oocyte. So we're going to make that right now. So about month 4, the primary oocyte, it needs protection. It doesn't have any protection, so we need to form a wall around it, and that's what happens first. So we get a new single layer of cells that form around it. And these cells are called follicular cells, or you could call this the follicular epithelium. And this surrounds and walls off our little primary oocyte. Okay, and now this is confusing. So now we have our primary oocyte is not naked and afraid anymore. At least it's got a little comforting wall around it. the primary oocyte and the follicular epithelium together take on a new name, and this is called the primordial follicle. Primordial follicle. So the primordial follicle is made up of the primary oocyte, stuck in prophase 1, and the follicular epithelium, that's the follicular cells that surround it. And this wall is going to get crazy big, but right now it's just one layer. Okay, so that's where we are. So primordial germ cells, we went to oogonia. Oogonia matured enough to become a primary oocyte. The oocyte went through meiosis, got stuck in prophase 1, but at least it got a wall around it, so now it's the primordial follicle. Primordial follicle is made up of the primary oocyte plus the follicular epithelium wall around it. So make some note cards, get that down. Okay. So the primordial follicles, they're kind of strong, but they're not that strong. It's only a single wall of protection. Anybody know who that is, by the way? Mr. Spock, right? The original Star Trek. Mr. Spock's not supposed to show emotion, right? It's very rare. He's half human, so he's got a little emotion that creeps out. So he reads this and he's like, oh my goodness, that's crazy. So by the 5th month of development, you have about 7 million primordial follicles, so everything's looking good. You should be able to reproduce like crazy with 7 million primordial follicles. But for whatever reason, they start to die off, and we don't quite understand the process of this. But we lose way more than half of these things by the time birth starts. So at birth, they go from 7 million down to 2 million, and we don't understand the mechanism behind that. And to make things worse, as the child ages, by the time they reach puberty, about 10, 11, 12-ish for biological females, that's called menarche, by the way, when they start getting their period, there's only 40,000 of them left. So they're still delicate, and it's probably because they only have that one layer around them. And they get exposed to maybe they take an airplane ride with a little kid and a little radiation, which doesn't bother, not enough to cause cancer, but maybe it's enough to knock off some of these, who knows. But there's not that many, there's still a lot. I mean, 40,000 is still a lot. And then out of these 40,000, only 400 of them ever really wake up in complete meiosis, and they get a chance to race. We're going to talk about the great race. And the dream of every one of these primordial follicles is to ovulate. And that's a rare accomplishment. Even rarer is to become fertilized by a sperm. I guess that's the ultimate dream of every one of these primordial follicles. So only 400 get a chance to wake up. The rest of them just kind of die off. Question?
When you're saying that, are you saying about like 400 menstrual cycles? Is that what you're saying?
Yeah, I guess, yeah, I never thought about it like that. Yeah, but there's one, every menstrual cycle they take turns. And remember, this is going on in both ovaries at the same time. So yeah, that would be about, they get a chance, so that would be correct. Okay, yep, so 99% of them, they don't even get a chance to race. And we'll talk about the race here in a second. All right, so here's the great race. The great race is called follicular genesis. So if you want to ovulate and get out of the ovary prison, you have to ovulate to break out of there. And so to do that, you have to go through a process called folliculogenesis. Folliculogenesis. And that's where the primordial follicle, they wake up and they race. So it happens at about the age of 12, again, when menarche starts. And each cycle, only 12 of them, or only about 20 of them, and this is variable from 10 to 30, so I've seen 50 sometimes, but on average, about 20 of these primordial follicles will start to grow, and the other ones don't. Just these 20 get a chance to race. One of these is going to ovulate out of these 20, and the race is on. The race is called folliculogenesis. So off we go. We have 20 of these waking up. What wakes them up? used to have slides on this, and there's all kinds of theories, and it seems like it changes every year, so we're not even going to worry about it. It's not in the board book, so you won't get a board question, but the bottom line is we're not really sure what wakes them up, and what wakes these 40, what are these 20 up. Why do 20 wake up and the other ones don't? It's incredibly complicated, and we don't even understand that yet. But every month in one ovary, 20 of them wake up. And out of these 20 racers that start the race, only 3 of them get fairly large. They get to 8 millimeters in size. So it's kind of like coming down the home stretch. There's 3 of these primordial follicles that get a chance to ovulate. The rest are going to kind of fade out and die away, as we'll see here. Okay. We do know what drives these racing primary oocytes and follicles, primordial follicles. It is some hormones, so it's FSH and LH that do it. So it's an excretion of these hormones each month that kind of starts off the race. So each month the hypothalamus releases something called gonadotropin releasing hormone from the hypothalamus. That goes down to the pituitary, the anterior pituitary, and stimulates a surge in LH and FSH. And those are, together you can call those the gonadotrophs or gonadotropins if you want. But specifically they're LH, luteinizing hormone, and follicle stimulating hormone. Okay, so I guess I jumped ahead. So growth hormone, or GnRH goes, stimulates the anterior pituitary. Anterior pituitary responds by releasing these gonadotropins. Who are the gonadotropins? That'd be a nice, easy question. Who are the gonadotropins? Which one of the following is not a member of the gonadotropins? So FSH, follicle stimulating hormone. And remember, on boards, do they write abbreviations on boards? No, they frown on that. So it'll always be spelled out. Follicle stimulating hormone, luteinizing hormone are the ones that are, those are the gonadotropins. Okay, so what drives their growth? Why do they growth, and how does this work? So these racing follicles have sprouted some receptors, and they appear only to be on the 20 that wake up. And it's believed that they all get a start, but what makes the top 3 pull away from the pack, and one theory is they just simply have more FSH receptors than the other ones. So the more FSH receptors you have, the more FSH combine to the cell and stimulate the growth of that cell. So that's the theory. And the theory is the one who actually wins has more FSH receptors than the other ones. It's probably more complicated than that. So arguably, so primary follicles change to a primary follicle. So arguably, under the influence of FSH, the primordial follicle gets big enough to become, to get a new name. Now it's just too big, so they call it the primary follicle at this point, which is confusing, I know, but this is embryology. The primary follicle, at least it contains the primary oocyte inside of it. So an AKA, sometimes these are called primary growing follicles, which makes sense because they're the ones that are growing and racing to see who gets the biggest, who gets to ovulate. And we're still in the process of follicular oogenesis. And primary follicle, remember, it still contains its little passenger. It's still only 1 cell thick, and it's the primary oocyte, which still lives inside this primary follicle. What's the difference between a primary follicle and a primordial follicle? Primary follicle are only seen in the ones that are racing to ovulate. It's bigger than the other ones. You can see these under electron microscope. You can tell the difference. All right, and we also remember that the primary oocyte is still stuck where? Prophase 1 of meiosis, or is it mitosis? Meiosis. All right, let's look at our racing primary follicle. So here's our racing follicle. We can sell it. It still has a single wall. These cells are getting bigger though, but it still has only a single wall of protection. There's our primary oocyte stuck in prophase 1. these cells, I forgot to mention that these cells, all cells have to sit on something, right? Cells are always sitting to a basement membrane or basal lamina. What happens when cells detach or don't have a basal lamina? What are they known as? Cancer. Cancer cells, to become a metastatic type disease, they have to break loose from the basal lamina. So thank goodness these are attached to a basal lamina. Okay. Follicular cells have morphed into a cuboidal shape. So these are simple cuboidal. Okay, they used to be squamous, now they've morphed. And yeah, that's another way to tell, where scientists can tell the difference between a primordial and primary follicle. If they look at the cells here, if they're square or cubed-like, then it's a primary follicle. If they're flat, squamous is flat, right? If it's really flat, it's kind of skin cell-like, then it's still a primordial follicle. All right, let's look at the zona pellucida, because that's important. So we're going to form that next. So let's meet the zona pellucida. So the racing primary follicles, they develop a new layer of cells around the primary oocyte. And this, once they morph into this cuboidal type, we get a new name for these things. So these will be called the zona pellucida. And this is important. We're going to go into the weeds on the stuff that these secrete, because these will follow the primary oocyte during ovulation. They go right out into the fallopian tube, unlike the rest of the stuff that is around this. So they're really important. So zona pellucida is going to be forming right here. It forms underneath these follicular cells. These follicular cells, the protective cells, are still there. But right now it's a membrane and it's starting to thicken. It's going to develop 3 glycoproteins here in a second. So not completely understood again how this all works. I used to go into the weeds on this, but it's not in board book, so I took that all out. But we do know that there's 3 types of proteins that are created to make up this zona pellucida. And this will be one of the protective cells right here. So they form underneath that one layer of protective cells. So there's zona pellucida 1, zona pellucida 2, zona pellucida 3. So these are the 3 layers of the zona pellucida. That would be a nice question, wouldn't it? Since I write board style questions where there's only 4 answer choices, this would be a perfect question for me to write. Which one of the following is not a building block of the zona pellucida? I can see the question now. ZP1, 2, 3, or 4, or 8, or PZ. I can make some crazy stuff up. So make sure you know these. Especially make sure you know ZP3, zona pellucida 3. And these are glycoproteins, by the way. Zona pellucida 3 is super important because that's where the sperm docks to. If you're trying to fertilize an egg, you have to have ZP3. There's another one that kind of works, but if the female has a gene mutation for the glycoprotein ZP3, probably will be infertile or have a very tough chance of becoming pregnant because of this. Just because of this little green one right there, ZP3. Okay, again, most important of everything I just said there. It's the most important because the sperm binds to it. ZP2, going into the weeds, ZP2 might help it a little bit, but ZP3 is the one that you want to remember. Okay, everything I said already. Sperm will have great trouble getting through. Oh, I didn't say this. This thing is tough. The sperm has a tough job getting through the zona pellucida. We'll learn about the corona radiata. Sperm can get through that no problem. Sperm have trouble getting through this, and we'll talk about that probably next time. All right, so even after fertilization, as I said, the zona pellucida stays around. It launches, it ovulates with the oocyte, and it goes inside mom's uterus with the oocyte, even the fertilized with the sperm inside of it. So it doesn't, we don't get rid of it until the fertilized egg drops down into the uterus. So it sticks around for a really long time, really protects that egg. Okay, look at the future, where are we going in the future? So just a little sneak preview. Here's a sperm head. And in order for the sperm to get into the egg, it can't penetrate through this zona pellucida without doing this. It has to bind to the ZP3 of the zona pellucida. And the sperm has a weird looking receptor, kind of looks like a bacteria almost. That's called SED1. So I can promise you that I'm going to test you on SED1 and ZP3. So make sure you know those two things, and that could be on boards as well. All right, and we'll get into that. There's a GAL-T, kind of a secondary docking station, but the SED1 is the one you want to know. Go Racers Go. Okay, so we're still growing. We're still trying to get big. So after the zona pellucida forms, that single layer of cells, the follicular cells, that's not enough to protect these cells that are getting so big. So we need to make that a multi-layer structure. So that's what happens next. So the follicular cells, those little cuboidal cells, they start splitting and splitting and splitting via mitosis. And pretty soon we don't have a single layer, we have double layer, and then a triple layer, and then 4 and 5 and 6. And it gets bigger and bigger, so we create a multi-layer structure. This is driven by FSH, I'll just say that. We won't get into the weeds on how that works. But our primary follicle is, it's really increasing in size at this point. In fact, here's what we have now. Look at our primary follicle. Instead of one layer of cells around the primary oocyte here and around the zona pellucida, we got what, like 6 layers here, 8 layers? I'm not going to count them, but we got a bunch of layers. Noteworthy too, look where the basal lamina is. That basement membrane of the original cells, it's pushed way out here, but that's important still. That's the original basal lamina or basement membrane pushed away from the primary oocyte. Okay, so about 8 layers deep at this point, we're going to get a name change. So once the follicular cells are 8 layers deep, the entire layer changes names, and now we're going to call this the stratum granulosum. Stratum granulosum. So all of this stuff, stratum granulosum. Okay, what makes up the stratum granulosum? So now these are just simply going to be called granulosa cells. Granulosa cells make up the stratum granulosum. There's the label right there. So yeah, we got a whole bunch of cells. And now the primary oocyte is happier. It's got much more protection around it, so it feels nice and safe. And yeah, some authors go AKA between follicular cells and granulosa cells. I won't do that though. Once it's thick enough, once it's 8 layers deep, I'm going to call these granulosa cells, like the board books do. Are we good so far? Good as possible? Brains are still hanging in there? Considering it's Friday morning, 7, what is it, 45?
It's 8 o'clock.
8 o'clock, okay. All right, new structure. You ready for a new structure? So now we need to talk about what happens outside this basement membrane. We got some new stuff that's forming out here. So right after this stratum granulosum forms, we got new stuff forming outside that basal lamina. And this is called the theca folliculi. Theca folliculi. So all this stuff, theca folliculi. And as that matures, it's going to split into 2 layers as well. So we have the theca interna, which means the inside layer, theca externa, which means the outside layer. So pretty easy there, but probably another word. Do you guys know that word? Do you do that in histology? Or is this all foreign material? Do you guys know what theca, have you ever heard theca folliculi, or is this new stuff?
I don't remember.
Don't remember. Oh, exciting, you learned something new then today. Theca folliculi, and there we go. So the cells differentiate. There's different hormones and different growth factors are expressed in the cells of each, and it makes them unique, but it's very important for the process of ovulation, as we'll see here in a little bit. So yep, theca interna, theca externa. Looks like I got two cells of the same, two slides the same. All right, let's look at the theca interna, means inside. So this one is highly vascular. It has a lot of blood vessels in it, which will be important later on. By the way, the term angiogenesis, that means blood vessel formation. So I used to go through how that happens, but I think that's too deep. I think we'll just say angiogenesis in this inner layer is very active, and it has quite a developed little blood vessel system that's going here. And some of the other cells in this area are fibroblasts, and collagen are also laid down here. So it gives it some substance. It can protect a little bit, but blood vessels are kind of the key here. A lot of the cells in this layer have also developed their own receptors to LH. We'll see how that comes into play here in a little bit. The theca externa, so think the interna, blood vessels, lots of blood vessels. The externa, it has a lot of smooth muscle fiber that developed. And what does smooth muscle like to do? It likes to contract, right? And so the contraction of these smooth muscles is actually will help kind of shoot the primary, which is going to become the secondary oocyte, out during ovulation. So this is a very important layer. Okay, so it gives the ability to contract, and the contraction of these muscle fibers helps eject the oocyte of the ovary at the time of ovulation. All right, let's meet another structure. Should we meet, or do you want to do a lap? Or should we meet? Should we keep going, or do you want to do a lap? Keep going, everybody's okay? Brains are still functioning? All right, let's go a little further. So what does antrum mean when you hear antrum? It's like a little hole or a little cave that's starting to, a little carved out area. And that's what happens next. We get the development of these little caves that start to form. They're filled with fluid. And the little caves start to coalesce together into bigger caves and bigger caves. And let's see what that looks like. So here we go. We're getting a bunch of little caves like popping up all through here. The artist didn't draw them in here, but they start coalescing together in one area. Now we got a pretty big little cavern right here, and that's called the antrum. The antrum. And we're going to see a story later on to that kind of a similar story to this. There's some, you remember the chorionic cavity? Chorionic cavity will be a very similar story as we get further along. But this is going to get bigger and bigger and bigger, and pretty soon this little lake, if you will, it's going to completely, not completely, but it's going to almost completely surround our primary oocyte. But there's going to be a little stalk right here that still connects it. Okay? But the formation of this antrum is important because we get another name change. So once this antrum, or this little lake, forms, now we're going to get the name secondary follicle occurs. Okay, some AKAs for that. Secondary growing follicle, antral follicle. I'm just going to call it secondary follicle. So, okay, confusing. Let's go back to our little timeline here. So primordial germ cells, they morphed into oogonia. They grew a little bigger and became the primary oocyte, which was naked and afraid. Primary oocyte got a wrapping layer around it of a single cell. They're squamous at first, and then it became the primordial follicle. Remember, that's a two-part structure. It has the primary oocyte within it, and then it has that little protective layer of squamous cells around it. Then they started racing, 20 of them, and became a primary follicle. Then those have just morphed into the secondary follicle. Why? Because we have developed this lake right here, this cavity, this antrum within the stratum granulosum. That's our little, where we're at. Okay, yep, so this just continues to get bigger and bigger and bigger, as I said, and it starts to kind of pinch, kind of separate the primary oocyte from the rest of the stuff. But it pinches off and creates a little stalk called the hillock, almost like the formation of the umbilical cord in a way, same kind of story. But this lake is also going to create two new layers because it's a lake, it's splitting. We have a south shore and a north shore when a lake occurs. So let's see what it looks like now. So now this antrum has gotten much bigger, but this area never splits off. And this is almost, will be like the umbilical cord. This will be the little human, a little bilaminar disc when we get to that, similar story. But it has created a north shore and a south shore, which of course have their own names that we need to look at. So let's see where I am. So the antrum of the secondary follicle has split into two layers, and let's name them. So we have the mural granulosa layer and the cumulus ophorus layer. What a weird word, cumulus ophorus. Good name for a dog maybe, cumulus ophorus, or maybe just ophorus. That'd be a good name. So these are the two layers, so let's look at them. The ones in kind of blue here that surround the primary oocyte, those are the cumulus ophorus. These are going to stick around as well after ovulation occurs. Remember, this one will be the zona pellucida. This will become the corona radiata. You're going to have to know that name. We don't call it that right now. It's still the cumulus ophorus, but that's a great question. The corona radiata came from what type of cell? Cumulus ophorus right here. All right, and then these are the mural granulosa cells. They're still the granulosa. I mean, these are all granulosa cells. They just got new names because they're in a new territory now. Are we good? Good as possible? Okay, we said that cells immediately are the cumulus ophorus. These will ultimately get ejected, as I just said. So this layer right here will go with, during ovulation, this layer goes with to protect the oocyte. This layer, the mural granulosa, stays behind. So this is not going to be ejected from the ovary. All right, yep, we said they're going to be renamed into the corona radiata. That's the first barrier. We'll look at that next time. Okay, so only one. So out of those three, who's this guy, by the way? Amazing athlete, Usain Bolt, incredibly fast. So just like the race, there's one that's going to pull away from the pack. Maybe not this far away from the pack. He's like messing around, isn't he? He's not even. He's like coasting at this point. Must be nice to be so talented you could just coast and win an Olympic medal. But anyway, one will pull away from the pack. And it'll get, and what I mean by that is they're not like physically racing. Their races grow in size. So one of them is going to grow bigger than all the other ones. And the one that grows bigger, kind of spoiler alert, that is going to be the one that's going to ovulate. The other ones are going to degenerate away. So how, but before we get to that story, how do the big three survive? What happens to the other? We said 20 are racing. What happens to them? Well, they die, and this is how this works. So the big ones are now so big that they can secrete estrogen. The other racers can't. They're not mature enough to do that. So once they can secrete estrogen, they really don't need FSH anymore. They become independent of FSH, and they do something really mean. Once they pull away from the pack, they secrete a hormone called inhibin. And inhibin will go up to the pituitary gland, and it'll inhibit. It'll shut off the production of FSH. So we get a little drop in FSH. And FSH is going to come back, but this little drop is enough to make the other ones, they can't compete anymore, they can't keep growing, and they just degenerate away. So now we only have three. They basically killed off the other ones. So the younger ones that haven't developed enough, we have to say goodbye to them. Okay, and one takes the lead. And now once Usain Bolt pulls away from the pack, you can start the stopwatch. We're going to start a 24-hour clock because within 24 hours, Usain Bolt's going to squirt right out of the ovary who's going to ovulate, and he's going to win the game. So the largest one, once it reaches a diameter of about 10 millimeters, it gets a brand new name to set it apart from the pack. It's called the Graphian follicle. Graphian follicle, make a note card. It's the largest one of these three racers. Sometimes it's called the mature follicle. Sometimes it's called the tertiary follicle. Fortunately, the two board books, one uses Graphian follicle, one uses tertiary follicle. So you should definitely know both of those, because I don't know which one of those will show up on board since they're both board books. Okay, and the Graphian follicle almost always is the one who ovulates. Why did I say almost always? Have we ever heard of twins before? Once in a while, it's like a photo finish, and the race is so close that they both ovulate. So that's a possibility. That's one way you can get twins. And the other, usually the ovary, one ovary releases the egg, the other one's resting, then the next month, the other, usually every other month, the ovaries are active. Every now and then, both ovaries go through this racing process. Another way to make twins. But most of the time, there's only one. And again, spoiler alert, within 24 hours, that Graphian follicle is going to shoot out the primary oocyte along with the zona pellucida and what will be the second layer up, and it's going to ovulate. Okay, I think we got everything there, and we're going to see how that process works right now. Okay, so coming down the home stretch, let's resume meiosis. So finally, we're going to wake up. So how exciting. So about day 14 of the menstrual cycle, the LH and the FSH, they surge. So that inhibit doesn't have any effect at that point, because we're going to get another surge. Larson calls this the ovarian surge, which I like. But that's going to really kind of push us over the top, or push that Graphian follicle over the top. The surge stimulates only the Graphian follicle, primary oocyte, because it's so far away from everybody else. So finally, meiosis resumes. Remember, we're stuck in prophase 1, so we're going to finally finish prophase 1, and we're going to go into metaphase. Yay, happy days. How does that work? It's luteinizing hormone, which they think is what finally stimulates and wakes things up. But don't get too excited yet. So, remember what meiosis 1 creates. We'll do a little flashback here. So meiosis 1, that's the reduction division. It's going to split the primary oocyte into two parts. A secondary oocyte, well, that's easy. But it's not like mitosis where you get two of the same thing. You get this weird little thing called a polar body. And a polar body, it can't live. It's similar, but it's really almost a dead cell, you can think of it as. It doesn't have much cytoplasm. The secondary oocyte stole its cytoplasm before splitting, so it's an unequal cytokinesis that occurs. But the key is to remember this is a reduction division. So the secondary oocyte now has only 23 chromosomes. There's no more pairs. It's not a diploid. It's a one end, which is like half a diploid or haploid. Okay, so that's the reduction division. Remember, the primary oocyte was 2n46 chromosomes. Now we just split that in half. Okay, so let's kind of look at the meiosis 1 and meiosis 2. So we started with primary germ cells. We went to oogonium. The oogonium finally started meiosis 1, started interphase. Everything was great through there. What happens in interphase, by the way? Test your brains. Interphase. Can we see the chromosomes in interphase? No. They're tiny. What are tiny, skinny chromosomes called? Chromatin. So chromatin is starting to thicken, but the DNA replicates. The chromatin replicates. So interphase is the replication phase. Because we're going to split things, we've got to have two strands of the DNA. So we get into prophase 1. Now we can see the DNA. They've thickened enough. And we go through prophase, crossover curve, all of a sudden we get stuck. But now, because of this gonadotropion surge, the Graafian follicle, the Usain Bolt, he's the only one who gets this privilege, he starts up again. So, yay. So we go to metaphase 1. We go through anaphase 1. We go through telophase 1. The cell splits, and the artist forgot about polar bodies, so I had to kind of draw this in for him or her. But we get a polar body which is much smaller, has no cytoplasm, and then we get our mature secondary oocyte, which is 1N23 chromosomes. Great. So we go into mitosis 2. Usain Bolt is sprinting. He's kicking back, and he's not paying attention to what's going on. And oh no, because he's not paying attention, he gets stuck again. That's really not what happens, but that is what happens. But this time we get stuck not in prophase 2, we get stuck in metaphase 2. So another fun fact to remember, Graafian follicle gets stuck with regard to meiosis where? And I could say meiosis prophase 1, meiosis telophase 1, meiosis prophase 2, meiosis metaphase 2. That's the answer. This is where it gets stuck. In fact, females, every time you have a period, this goes out. This secondary oocyte stuck in metaphase 2 goes right on out. It doesn't pass. If you get pregnant, if a sperm gets in here, and you still might go on out. But if you implant into the uterus wall and you get taken inside the uterus, finally you will go through meiosis 2, anaphase 2, telophase 2 cytokinesis. So that excitement is coming soon. I know you can't wait. So yeah, so we are stuck again. So after going through prophase 2, meiosis 2, Graafian follicle gets stuck again in metaphase 2. So bottom take-home message, meiosis never finishes in this process of forming the egg, unless fertilization occurs, and I could have added unless implantation occurs. Okay, and yep, it'll stay this way until the sperm fertilizes it. So stuck again. Definitely at the halfway point. Break, or you want to push through all this? Yeah, I got some break, some shaking. This loud. Okay, timeout, break, lap. Get those calf muscle pumps working. What do the calf muscle pumps do? How do you get blood out of your lower extremities? By moving your calves. Your soleuses are the calf muscle pumps. Go use your calf muscle pumps.
Fun fact for the day.
Another fun fact. So many fun facts today.
There's a lot going on.
You have a job. What am I doing? You're going to remind me to turn that on. Just remind me.
It's fine.
Hopefully I
Fine, okay.
Okay.
How are you? Good. You know, I realized that I didn't even tell you guys.
All right, here we go. Get away from the moose jaw. Oh, that's right, you can't see my laptop. All right, what happens next? I know you're just can't wait to see what happens next. So, also on day 14, so if we're going to release this thing, so here's our secondary oocyte. This is your egg. We got to break it free. It's got this attachment on it. So we got to break that loose, and that happens about on day 14. So that, remember, that's that little anchoring structure that's called the hillock of the cumulus operus. That breaks down, and now the secondary oocyte is loose in the antrum. It's floating around in here. Okay, so what happens next? So this, it's got to break out. It's got to get out of all this stuff. So we've got to figure out a way for this to get out. And then it's got to get out through the uterine wall as well. So we got a lot of stuff to get out of. So the first thing it does is some of these cumulus ophorous cells, they start secreting a hyaluronic acid rich matrix. So they start making hyaluronic acid molecules. And if you remember from disc anatomy, those love water. They're very, is that hydrophilic or hydrophobic if you love water, if you love to hook up with water? Hydrophilic, so they're very hydrophilic. So these cells start spitting out these hyaluronic acid molecules. They start grabbing water, and water can start diffusing in here just like any cell. And it starts to expand because now we're getting more and more water molecules drawn in. And so everything starts stretching out. So hyaluronic acid, sometimes that's called hyaluronin, kind of an AKA for it, hyaluronic acid or hyaluronin, binds water like crazy. And as I said, things are going to swell like crazy. And that's going to start to put some pressure. In fact, it almost doubles the size of the graphene follicle. It increases up to 20 millimeters. So now it's way ahead of all the others. It's just huge. So the secondary oocyte and the mass the pressure of it getting that big starts to put pressure on the follicle and the ovarian wall as well. Because by this time it's docked right against the ovarian wall, and it's starting to put so much pressure that the ovarian wall is starting to get a bump on it. Larson calls this the cumulus expansion phase. Okay, now hatching. So let's talk about hatching. We're finally going to get out of here. So we got to get out of the follicle. The follicular wall must be broken, and that pressure that's increasing is not quite enough to break it. So another thing happens. So remember the theca folliculi? That has a whole bunch of blood vessels, we said. Theca folliculi blood vessels. They start, the epithelial cells or endothelial cells of these blood vessels start releasing prostaglandins and histamine. Prostaglandins, what is that? When I say prostaglandin, what pops up in your brain? It starts with an I. Inflammation. And so these are it's like gasoline. They start an inflammation that starts to weaken the graphene follicle wall, and because it's stuck to the ovarian wall, the inflammation starts weakening and inflaming that wall as well. And that's why some females, they get a little pain with menstruation because of this inflammation of the ovary. It hurts. That's a highly sensitive, it's got a lot of nociceptive fiber in it, and that's one irritant type of thing. So we're stuck against the ovarian wall, we said. It's stuck so bad it looks like a bump, and we can even see it if you stick an endoscope in there and look at the ovary right before ovulation. You can see a bump on there, and we're going to look at that here in a second. And there it is. So there's the ovary, and you can see this bump. This ovary is it's about ready to ovulate. So we've got an inflammation going on here. We've got prostaglandins being secreted. We got this thing filling up with water and expanding, and it's putting a visible bump right here. And that has a name, of course, this visible bump on an ovary that's getting ready to ovulate. That's called the stigmata. Stigmata, or the follicular stigmata. So make sure you know that word. Okay, shoot it out. So because the wall is weakening, because of inflammation, the ovarian wall is going to rupture really soon. Once the wall ruptures, because of the building pressure and the inflammation, the graphene follicle will shoot the secondary oocyte out because of all that pressure. But it's important to know that the graphene follicle itself does not ovulate. The only thing that ovulates is that secondary oocyte at this point, and the layers around it, the two layers around it. Okay, so a little bit more in depth with regard to the theca externa. So two more actions we can talk about. They both come from the theca externa. That's that very outer layer of the graphene follicle. So Remember I said that outer layer, the theca externa, is not filled with blood vessels. That's the one filled with smooth muscle. So the smooth muscles start accumulating calcium. I used to go into a big thing. I took all those slides out. It's getting too deep. But remember what happens when calcium goes inside of a smooth muscle cell or a skeletal muscle cell. It stimulates contraction, and that's exactly what happens. It starts contracting and squeezing down on the secondary oocyte and attempts to make not much room for it. And so it pushes into the uterine wall very, very hard because of that. Okay, so it weakens the follicular and ovarian walls. So that's just yet another. So we got inflammation, we got contraction of the smooth muscle, we got a swelling inside going on as it accumulates water. So those are three forces that are pushing our little egg out of there. And yeah, once it finally squirts out, the secondary oocyte is outside the ovary, the race is over, you have ovulation. Ovulation has occurred. Then what happens? So now we got the egg on the outside of the ovary. That's where these, you remember these from anatomy, the fimbriae come in. The little fingers are kind of hanging like an octopus hanging over the ovary here. And they can shift around, they can move. They will sense where the stigmata occurs and they will kind of shift themselves to get ready to catch it. So it's important that they catch it, and they do catch it, and they literally sweep it up inside of the fallopian tube. Okay. So yeah, is it possible that this ovary could get loose and escape these fimbriae? Yeah, have you heard of an ectopic pregnancy? We'll look at the different types of ectopic pregnancy. If the fimbriae miss, this thing could be floating around out here in the pelvic cavity. It could stick on the ovary. It could stick on the internal abdominal wall. So it's possible. It doesn't happen that often. But could a sperm actually, if it was stuck right here on the ovary, could a sperm find it? Sperm could actually find it. Those sperm are so powerful swimmers. They can swim right out of the fallopian tube looking for it. They can swim into the abdominal cavity and they can find it. And you can have babies sometimes that are created on the abdominal wall they grow because of this. It's rare, but it can happen. So we'll look at that more when the time comes. Okay, so what happens to our Graafian follicle now that it's shot out its secondary oocyte? Does it just degenerate away? Not really. It's still got some work to do. So the cells of the corpus luteum, they all of a sudden start dividing again through mitosis, not meiosis. And they morph into a new cell type, which are called luteal cells. These are endocrine cells. Okay, so just because its baby, so to speak, is gone, its secondary oocyte has been shot out, its work is not over. These cells morph into luteal cells, and they start to fill up the broken down Graafian follicle. And once they fill up with these new cells, the Graafian follicle gets a new name. It changes to the corpus luteum. This is high yield board stuff right here. Graafian follicle becomes blank. 4 answer choices. After ovulation, the Graafian follicle morphs into blank. I can see my question coming. And it's the corpus luteum. You've got to know that word. And the corpus luteum is made up of what type of cells? Is it made up of corpus luteal cells? No. Graafian follicle cells? No. Follicular cells? No. It's made up of luteal cells. Luteal cells are endocrine cells, and they secrete some hormones that we're going to look at. Okay, so what does this corpus luteum does? It's an endocrine structure. It secretes hormones. Where do hormones go, by the way? Into the bloodstream? Yep, they go into the bloodstream. They secrete estrogen and progesterone. And we're going to look at something called, have you heard of HCG, human chorionic gonadotropin, that's going to come into the play here. Because that's going to bind to these cells and prolong their ability to secrete estrogen and progesterone. But for right now, they can do it all by themselves. And this progesterone is very important for preparing the lining of the uterus, the endometrium, to get ready to receive the secondary oocyte should fertilization happen to occur. So super, super important. If the egg doesn't get fertilized, then the corpus luteum will stop. It only does this for a couple days. And if HCG doesn't bind to it, HCG only occurs when pregnancy, when fertilization occurs, right? That's the early pregnancy test uses HCG, one of them uses HCG. And if HCG isn't around, then this corpus luteum starts to wither and die away. And here's another high yield board question, noteworthy card. What does the corpus luteum morph into? It morphs into this scarred up structure called the corpus albicans. Corpus albicans. Can the corpus albicans secrete estrogen and progesterone? No, it's basically a dead sack of cells. So it does nothing. It does nothing. It does come out. The fimbriae grab it though. It is ejected. The fimbriae can grab it and it's expelled during the female's period, or menses, that's called. Okay, so corpus luteum, dead Graafian follicle, which doesn't work anymore. All right, just a few more slides. Let's meet the ovulated oocyte that we just spent so much time making. So there it is. There's kind of what it looks like. That's the cumulus ophorus layer, which is, I don't know if it looks like the sun, but let's go through it. So here it is. This is what we worked so hard to build. It's haploid, 23 chromosomes, basic softball questions. It's called a secondary oocyte. It's stuck in metaphase. Which metaphase, 1 or 2? Metaphase 2 we made it through, right? Because we had to go all the way through metaphase 1. We had to go through the entire meiosis 1 because we got a haploid. We made a polar body and we made a secondary oocyte. Stuck in metaphase 2. It's normally swept up by the fimbriae and a little anatomy. It's swept up into the infundibulum of the fallopian tube. That's the very end. The fimbriae come off the infundibulum, so good to know. I do like my anatomy. And there it is, stuck in metaphase 2 of meiosis, we said. But it has, where's the oocyte? Where's the secondary oocyte? Where's the DNA? Where's the nucleus? It's deep inside this thing. It's protected. So the sperm has to get through all these layers. In fact, there's 3 layers. Anytime you see something with 3, almost guaranteed a question's coming. Really easy for me to make a multiple choice question like that. But it needs to get through these 3 barriers. I can see the question now. All of the following are barriers of the oocyte except for blank. So corona radiata, these are in order. The zona pellucida, that's a tough challenge. And then we have the oocyte's plasma membrane, which confusingly they call it the vitelline membrane, or vitalin, tomatoes, vitalin membrane. So we need to get through those layers. So here they are. This should be about 3 layers thick, and the author just drew maybe 2 layers here, but if this is the sun, this is the corona radiata, an eclipse, that part that glows if you look at it. When the sun is completely blocked, this kind of sticks out. That's where that came from, corona radiata. Then we have the zona pellucida right here, 3 layers thick. It's got those ZP3, 1, 2, and 3 proteins. And then the cell membrane itself, or the plasma membrane, or the vitalin membrane. So many AKAs. You think embryology is hard enough without all these AKAs. Nevertheless, there it is. Where's the DNA? Inside the nucleus? Yeah, that's where. And what's the job of fertilization? To get the sperm nucleus in here and to merge with the nucleus of the female. And that's what we're going to talk about next time. What are the 2 weapons that the sperm has to cut through these layers? It's not an easy job to get the sperm in here. It has 2 weapons. They're called the process of capacitation and the acrosome reaction. Another high yield board stuff here, especially the acrosome reaction. We got to get into the weeds. I think your brains have had enough for today. More importantly, the bird of the day. So this is a northern harrier. So let's do a practice extra credit question. Is that a robin? Is that a bluebird? Is that a bald eagle? Is that a coyote? I saw a coyote on my day off yesterday, howling like crazy. Got video of him just, I thought it was like a pack of them, one single coyote. I never knew they could make so much noise. But that is a northern harrier. I don't see those very often. They're down by the swamps more than out in the fields. All right, so that is it. I'm up here for questions. Come on up if you have questions. Use this time to study. I'm actually going to go after my questions are over. I'm going to go back to my office, and would you like some practice questions? Would that help you? I'm going to make some practice questions. All right. So we'll see you. If you don't have any questions, we'll see you next week. Give me emails, day, noon, night, Sundays. I always answer my emails.
You can take a flashcard and your slide deck. Is that a good way to start? Just do it. This is going to really have vocabulary. Yeah, I'm
I was too. When I graduated.
Oh, like I had to, because I just finished my first year. I had to throw out. I had to throw out my first quarter, like last couple of quarters, because it was just like overwhelming.
But they have electronic ones now, don't they?
I'm 45.
I like the old 3x5. I was like, I could not work without flashcards. I don't know what I'd do without it.
I'm the same way. I just hammer it. That way you can take it with you wherever you go. Say I'm on the coffee. Study.
It's good to know flashcard people are still around.
We're still around. We're all around.
Like do you use flashcards?
I use Quizlet or I don't really handwrite it. I usually just kind of type it out. But however, writing makes you learn it a lot.
Yeah.
Now it's just like a bunch of repetition. You're like, easy to figure out. Yeah. Okay, you said something about fika. I had just kind of like spaced out right here.
Fika.
I didn't get the definition.
Let's go back. That's that outer layer.
Right.
There we go. So that's just this last outer layer that forms. So we think it comes from actually cells in the basement membrane, which makes this, but it does appear, and it starts getting thicker and thicker, and as it grows, it differentiates into two layers.
That's why I'm always
The inside and the outside. Fika interna is on the inside. Fika externa is on the outside. The key is, for ovulation, is you have to have this smooth muscle. See all these little smooth muscle cells?
Yeah.
So those are contract. And there's an area that they move away, the smooth muscle migrates away, and they all contract here to literally shoot that down.
Oh, so the fika is the fika the smooth muscle or is the
Fika externa contains the smooth muscle.
Got it.
There's other stuff. Yeah, there's collagen in there. There's blood vessels.
Yeah.
Fika interna, that's just filled with, they don't even show them, it's just filled with blood vessels.
Gotcha.
And it doesn't really it doesn't squeeze it.
Gotcha. It's just kind of supplying.
Yeah, keeping the little creature alive. Primary follicle alive.
Thank you so much.
Yeah, no problem.
I just was wondering what the hormone was for the pregnancy test.
Human chorionic gonadotropin, HCG.
Okay, I did hear you right. Thank you so much.
Yeah, so we will not escape that. It's
Okay.
coming, probably next time.
Cool.
We'll talk about it.
Thank you.
Yes. So remember the sun picture? You good with that? Sun.
Oh yeah. It's the best. That woman is so smart.
There we go. So cumulus ophis here, that shoots out during ovulation. That goes right down the fallopian tube. And it's the same layer, just it's an Asian shape. So it becomes the core. It's the same cells. They just they want you to learn more.
Thank you so much.
Sure. Did my battery survive?
Oh, there's still a red light flashing.
There is, that's a good sign. Yes, it is alive. I have my